Everyone, let's begin cycle 5 on Mendelian inheritance. But before we start lecture, I have several announcements. First is that this is my final cycle that I'll teach to you this term. Cycles 6 through 11 will be taught by Dr. Beth McDougall-Shackleton starting Wednesday, October 15th. So there is no class on Monday, October 13th, due to Thanksgiving, and so Tuesday's class is canceled as well. Monday, Tuesday, no class. Cycle 6 starts Wednesday, October 15th. I will still be holding office hours all throughout and including October 16th, even though I'm not teaching that week. Your midterm exam is coming up on the 18th, so I will host the office hours that week to help you with any questions you have. So please come Thursdays, 1 to 2:30 in Middlesex College 105B this Thursday or the Thursday after. So a reminder that your midterm exam will take place on Saturday, October 18th from 2 to 4 p.m. This tab is now open and available on Brightspace for you to go and have a look. Please review all the instructions carefully. It is absolutely your responsibility to be familiar with all the policies that surround the midterm exam so that you arrive fully prepared. The onus is on you to make sure that you understand what the exam will entail and what to expect. You do not want to have any surprises on the midterm. The last thing you want to do is arrive and be surprised with something. So understand what you can bring, what you can't, and how the structure will be, what you're supposed to do, what you're not supposed to do. It's all laid out there for you. Practice exam, it says here, will be posted, but it already is posted on there. The practice exam is the exact copy of the exam from last year that your peers wrote. So use this as a great tool to practice. I would strongly recommend that you take this practice exam and you do this practice exam under testing conditions. So take 2 hours aside, set the timer, and do the exam from start to finish. And commit to answers. Don't say, oh, I'll come back to this and check it. No, commit. So you can grade yourself in the end and understand where you sit so that you can get the help that you need. I would recommend you do this exam in advance so that you can come to our Q&A sessions, you can go to the Biology Mentor Review sessions, you can go to office hours and have the opportunity to ask all the questions that you need. If you do it too close to the end of the week, then you may not have opportunities to ask questions. If you do need to take a washroom break while you're writing this practice midterm, make sure it is coming from that 2-hour allotted time. 2 hours is when the timer stops. Ask yourself and grade yourself, and that will give you really a good indication of where you stand and what gaps you need to fill. So speaking of the midterm exam, I have a wonderful former first-year biology student here with us today. His name is Daniel Bender. He was in your seat a few years ago, and he has some interesting and very insightful advice for you.
Can everyone hear me in the back okay? Yeah? All right. Good morning, everyone. So my name is Daniel Bender. I'm a third-year kinesiology student here at Western. So obviously you guys have your midterm coming up in about 11 days, and I wanted to tell all of you about my experience with this course, but in particular this midterm. So leading up to the midterm, I was still figuring out how to study for university-level exams. I had my biology and chemistry exams back-to-back, and I did not plan or allocate sufficient time to study for biology. The morning of the exam, I was still learning content that I had never really understood before, which is not something I recommend you guys doing. After writing the exam, I had that gut feeling I knew it hadn't gone well. Fast forward a few weeks, I'm sitting in chemistry class, and everyone gets the notification, exam marks released. So I open my phone, and I look down, and I see 17.7 out of 42. And I'll save all of you the math, that translates to 42%. I knew the exam went poorly, but I didn't expect such a low mark. Coming into Western, I had over a 97% average, and I was the recipient of the Western Scholarship of Excellence. I had never failed any test or exam in my life up until this midterm. I was really disappointed in myself and super upset, and I didn't know what to do. Later that day, I went to Dr. Schran's office and met with her. I explained my situation, and she told me I have 2 options. One, I can let this mark define me as a student, and I can dwell on it. Or I could say, yeah, you know what, I got 42%, but I can still finish with an A in this course if I change how I study and work even harder. Luckily for me and luckily for all of you, this course offers alternative grading, and I encourage you guys to experiment with the calculator to see just how poorly you can perform on the midterm and still finish with an A. Dr. Sharan was extremely helpful, and I met with her and the other instructors multiple times throughout the semester. There are many valuable lessons I learned from this course, and I want to share the 3 most important ones with you. First, university science exams do not only ask basic recall questions. You have to have a very deep and thorough understanding of the material to answer the challenging application-style questions correctly. Second is to attend the office hours regularly. Your professors all want you to succeed and are willing to help you achieve your goals, but you have to take the time out of your day to go to those office hours. And my last piece of advice is you have to learn from your failures. Otherwise, that's all it was, a fail. But learning from your failures, that is a success. Since completing this course, I managed to finish with an 80 in this course, and then I went on to take Bio 1002, Cell Biology 2382, and Biochem 2280, which I finished all above 90% in. And I credit all of that success to this first initial failure on the midterm. Thank you all for listening, and good luck.
Thank you so much, Daniel. And I just want you to know that it was very kind for Daniel to come and share this personal experience, but keep in mind that, this is not a unique case. Many students, in a class of over 2,000 students, many students are in that position. So understand that you have time to recover. We have alternate grading, and again, don't let grades define you. So what I want to talk about next is the Cycle 4 quiz. This is a question that was given to you. The correct answer is all of 1, 2, 3, and 4 are correct. But if you put 1, 2, and 3, I will also give you full marks. And the reason for that is because I did not clarify in lecture that DNA polymerase still has to use, or still can use, and has the ability to use, is 3' to 5' exonuclease activity during the mismatch repair process. Imagine the enzymes come, they cut their incorrect strand, and they remove it, right? Now DNA polymerase is going to come, and it's going to synthesize that gap. Well, it's not going to lose its 3 to 5 exonuclease activity. It can still move backwards if it creates a mismatch during that repair. Does that make sense? If you're driving a car and if you're going long distance versus short distance, you're not going to lose your reverse gear all of a sudden. You're still going to have that reverse gear to do it. But because this was not explicitly mentioned in class, I think a lot of students put 1, 2, and 3, thinking that 3 to 5 exo was reserved for proofreading only, and that was a very separate process. It is, but proofreading is still happening during the repair. All right, when it's filling it in, it can go backwards. It's not going to say, oh, I did a mismatch, but since I'm repairing, I'm going to let this one go. It's going to have to go back and fix it. The second one is this one here. This is not that much of a big deal. The correct answer is 1 and 3, which was straightforward for many students. But for the first 50 students that wrote the exam, there was a default answer correctly written as 4 only. That is incorrect. The correct answer is 1 and 3. So if you put 1 and 3 and you still got it wrong, that means you were in the first 50 students to write the exam. I will make that correction. I will have to go in and manually fix that. So I appreciate your patience. I won't return Cycle 4 quiz grades until much later. This week is a little bit stressful for me, and next week as well. I need to prepare for this midterm. So it's taking up a lot of my time, and I appreciate your patience, but I will get this mark out to you. The next thing I'm going to talk to you about is just your mental wellness and your mental being in your undergrad. Your mental health and your overall wellness is essential to your success as an undergraduate student. I've been doing this for 20 years, and I can tell you I have seen students that are absolutely brilliant, just at the genius level, and they're not able to perform at their full capacity because of their state of mind, their mental state. They're just not in the right state of mind to be able to perform at their highest. And you need to be able to do that, you guys, because it's so competitive now. You have to be in your A game all the time. And for you to do that, you need to be in the right state of mind. So the first year biology instructional team, we are committed to ensuring that you have access and guidance and all the support you need to thrive at Western. So the next following slides They highlight valuable resources that you can turn to whenever you need support, helping you navigate your academic journey with confidence and with care. These resources are available to faculty, staff, and students at Western. They say that they're free for all of us, but we all know they're not free. We're all paying into it, right? You are paying tuition, which is why you have access to it. This is take advantage of this. It's all there for you. Once you graduate and you're no longer paying tuition, these services are not available to you. So if the help is there, use it if you need it. If you don't need it, you don't need to use it. This is the stress continuum. This simply means that you can be anywhere in this continuum at any time, at any given day, any given month, week, year, whatever the situation is for you for that particular time. And this is continuous, but it's also reversible. It's forward and reverse. You don't have to be in a very specific spot all the time. It's very natural to move this way and then to revert back. And as long as you're not swaying back and forth constantly, that can take a lot of toll on your mental health. Try your best to look for the signs that tell you that you need to revert back. Early prognosis, looking at things early and finding it early. Prevention is the secret. Prevention is the key. The further you go to the right, the more difficult or more time it will take for you to revert back to the orange and the yellow and the green. So try your best to keep yourself in the green and get back to green as fast as you can. These are the common concerns for undergraduate students. This is proven through research. Did you guys know that university students are, by research, shown that most stressed people in society right now? I'm looking at the most stressed group in society right now. It's you guys. And so we understand that these are common factors, that it could be a mix of these. It could just be one thing you're dealing with, but imagine all the things you have to deal with and to focus at school. It can be very difficult to turn to help for this. And understand that there's mental wellness, mental distress, mental illness. We can't have, in 2025, having this as a taboo. Back in the day, guys, if you discuss your mental health, it was taboo. You don't discuss mental health. But it shouldn't be like that now, right? If you need the help, you guys are in medical sciences, health science, biology, in life sciences. You should be the one advocating and saying, yes, mental health is important. And we need to really be there and have supporting systems. This is the number you can call to make an appointment with any mental health counselor. They're a live person. This is not just a phone call, some Zoom call or someone just on the phone. This is physically someone that will meet with you. You can book an appointment. These are the counseling hours. They're trained, they're professionals, and get the help. It doesn't mean that you have to have something going on to go get, you have to be in the red zone. You can be in the green zone and walk over and say, how can I prevent myself from getting in the red zone? What are some things I need to look out for? I don't want to get there. I don't want to be there. So what can I do to prevent that? This is what I'm struggling with. Stress will always be there, everyone. Stress will always be there. The question is, how are you going to cope with the stress? That's what's going to determine your success, your ability to cope with the stressors that are there. You cannot get rid of stress in life. These are on-campus and off-campus resources. For example, Good to Talk. This is a confidential 24-hour helpline supporting post-secondary students in Ontario. You can call that number, and their expertise is to help post-secondary students. So you don't have to explain to them your whole situation. Well, here's how university works. They know. Just boom, get right to the issue that you're dealing with. Again, a plethora of resources. Use it to your advantage. Use it to your advantage. If you need it, it's there for you. This is a wellness navigator. If you go to this site, then you can actually plot your mental health status. You can just answer some questions confidentially. It's just for you to see. Nobody else is going to see this. And you just plot the numbers. These numbers can be whatever you want them to be. They don't have to be sitting at 100. Wherever you need to be, you just gauge and monitor. You can see the curves, right? You can monitor weekly, daily, halfway through the term. What is my mental status like? And when you plot something like this, you can ask yourself, oh, wait a second, that happened in September, October. What was happening during that week? Oh, I know. You might see a pattern. Whenever I have these stimulus in my life, then I seem to have my mental health go down. Maybe I need to get rid of these things in my life or these people in my life. Whatever you have to do, you can gauge it better. You have a better perspective. when you can plot something like this? Let's start lecture. We're going to finish off cycle 4 before we start cycle 5. This is an important slide. I wanted to take my time to go over this. This is a concept that's a little bit upper level, but every time we put something challenging for students, every single year students step up to the challenge and you just blow it out of the ballpark. So congratulations on that. So this is a concept that I think you'll really appreciate in upper years. We talked about transposable elements, and they are silenced in our body. We silence them. We have silencing mechanisms. And if they do jump, they have a tendency to jump in safe havens, so between genes or within the introns of a gene. So it's safe. It's not interfering with the product of that gene, whether it's RNA or protein. It's not interfering with the function of that product. However, if the TE lands in a protein coding gene and it does interfere with the expression, then you can have deleterious effects. An example is hemophilia. Hemophilia is an X-linked recessive disorder. So this gene that is affected is called factor VIII gene. It is a gene that produces a glycoprotein that is involved in the process of clotting your blood. So ultimately it makes fibrin, and fibrin is that factor that is involved in helping your blood clot when you bleed. So in hemophilia, people that have hemophilia, they will bleed, they can bleed profusely after surgery, after an injury, or even just spontaneously in their joints, the spontaneous bleeding. And the reason for that is because they don't have a functional factor VIII, because there's a mutation. So this gene is again in the X chromosome. So women can be carriers or have hemophilia, and men can either have hemophilia or not have hemophilia. They're not carriers. The mutation in factor VIII can be insertion, inversion, deletion, but another type of mutation is transposition. You can get a transposition event. A common TE that goes into that gene is known as line 1. So L1 is another type of transposable element that we have, in addition to ALU. When transposition occurs in factor VIII, you don't get a functional factor VIII gene, which means that you can't produce that ultimate fibrin that is required for clotting. So it's interesting because there are situations, in many cases reported, where the mother is not a carrier, not a hemophiliac, not a carrier either, and the dad's not a hemophiliac. But the child has hemophilia. So this is something that explains, and when you look at the genotype, you can see line 1 transposition in the factor VIII gene, which is indicative of non-familial hemophilia. You might be saying to yourself, wow, what bad luck that this person has line 1 transposition in their factor VIII genes. What are the chances in a 3.2 billion base pair region, right? But here's the thing. If you understand the evolution of transposable elements, they've jumped through evolutionary time, and they've landed themselves in safe havens in our genome. They're in between genes. They're in between introns within the, that's not coincidental, right? Through evolutionary time, we're now here as a species because our TEs are silenced and they're not interfering. So even though they're spontaneously, jumping, they're not quite that spontaneous. They do jump into spots that are, through evolutionary time, predetermined. They have spots that they like. So the factor VIII gene is a very long gene. It's 120 megabases, roughly. So it's a very long gene. On top of that, it has repetitive sequences within that gene. And TEs seem to like repetitive sequences because they themselves are repetitive, right? If they go into one another in tandem, they can be repetitive too. So they're like, oh, maybe my friend is over there in factor VIII. Maybe I'll jump over there. So it's not completely random. There is some rhyme and reason, which is the reason why you see certain genes being more vulnerable to transposition. It's because of their makeup, their sequence, and their general nature of what they look like. They're more attractive to TEs. So that's how you get a disease when you get transposition. You can also get gene shuffling by the process of transposition, and this is a really interesting upper-level concept that I want you to know in first year. To understand gene shuffling, what that simply means is that that gene that is in a specific locus in your genome, right, on a specific chromosome, will get moved or translocated to another region. It can be in the same chromosome or maybe even a different chromosome. and that can be carried by the TE itself. So when it undergoes transposition, it takes the gene with it. That's scary yet cool. It depends on if it's working for you or working against you. So to understand gene shuffling, there are a couple of things you need to understand. Genes are under the control of a promoter. So when a gene is expressed, gene expression means that the gene will be transcribed into RNA. It can either function as RNA or it can be converted to a protein if it's a protein coding gene. So transcription of a gene means that the gene is now expressed. So gene expression is something you will hear all the time throughout your life in life sciences. Gene expression. So these genes are expressed due to this promoter. What controls the promoter? Transcription factors. Factors that come and bind to the promoter and say, okay, transcribe the gene now. What's a good example you learned? You learned P53. P53 is a transcription factor that binds to the promoter of P21 and activates P21. So there you go. That's just a beautiful example that you've already learned. P53 driven P21 expression. So P53 can bind here, but there are a whole bunch of transcription factors. So each promoter has a very specific gene that it transcribes. The second thing you need to know is that certain genes are transcribed often and others not so often. So the rate of transcription depends on the type of gene. In other words, do you need this protein often in the cell or seldom? So for example, housekeeping genes, those that are involved in transcription and translation, for example, need to be made all the time. So ribosomes, tRNA, rRNA, RNA polymerase, these things are always there, so they're housekeeping. But then there are genes that are not housekeeping, like growth factors. Growth factors should not be there all the time, because then you'll get cancer. You don't want the cells to be growing uncontrollably, right? A lot of cell cycle regulators should not be active when they shouldn't be, et cetera. So there are genes that are housekeeping and those that are not. The third thing you need to know to understand gene shuffling is what happens when a TE undergoes transposition, i.e. a cut and paste. It can be copy and paste or cut and paste. So if it undergoes a cut and paste, when the TE leaves, it can leave part of its sequence in the gene itself. So the TE will move, but a part of its sequence will still remain. The second thing that can happen is that the TE can take part of the gene with it. Not all, but part of it. And the third thing that can happen is that the TE undergoes transposition and takes the entire gene with it. So here's an example of it taking the entire gene with it. Now gene 1, that was originally under the control of promoter 1, which is its proper promoter, is now under the control of the wrong promoter. Now this promoter could be a housekeeping promoter that is synthesizing housekeeping genes. But this gene is not housekeeping. So if it's a growth factor, for example, then it can lead to cancer. If it is a protein that is absolutely not supposed to be expressed in the cell cycle and drive cell cycle, it can lead to cancer. So you guys understand, you can think of examples of a transposition event that could lead to a disease. But this is the general concept. People that learn this concept that are now in third year do come to me and say, thank you for explaining that. That was very helpful, because that's just the start of a lot of other complicated concepts. Okay, so now let's turn to cycle 5. Cycle 5 on Mendelian inheritance. We're going to be focusing on one big concept: how phenotypic expression is a result of genotypic expression. Done. That's the summary of the entire cycle. Phenotypic expression is a result of genotypic expression. In other words, how do different genes or different alleles interact with one another to produce a specific phenotype? That could be another way to summarize cycle 5. How is it that these genes are interacting with one another? That's their genotypic expression. And how is that leading to this specific phenotype? The phenotype can be something you can see directly: size, color, shape, etc. Or it can be a phenotype you can't see. It's inside the cell. That can be also a phenotype. All right, so Gregor Mendel was one of the first to pioneer this concept, genotype to phenotype, and he used the pea plant to do this. And the reason he used peas is because A, it's very easy to grow, it has a fast generation time, but more importantly, he used pea plants because he could see the phenotype with his naked eye. In the 1800s, you didn't have scientific tools to dive deep into the nucleus and look at what's happening with the DNA, look at the molecules, look at the cell even. I mean, they didn't even know the DNA existed, let alone looking at these tools, right? So he had to be able to look at round peas, shriveled peas, green peas, yellow peas directly with his naked eye. So that was another reason why he chose peas. So peas was an organism chosen for that reason. And what he concluded were many things, but one of the things he concluded is that you need two alleles to manifest a specific phenotype. One could be dominant and the other recessive. So in this case here, big R on this specific locus, little r on this locus. And you can see that big R produces round, little r produces shriveled. This is a heterozygote, and the phenotype of this heterozygote is round because R is recessive. So the phenotype of the recessive allele is masked by the dominant allele, which is the reason why you see the phenotype of the dominant allele, because the recessive one is being masked. That's why it's called dominant. However, in this generation, sure, it's being masked, but if you take two heterozygote individuals or two heterozygote plants and cross them, then you can predict the probability that you will see the recessive phenotype in the next generation. It may not happen, but you can predict the probability. So that would be through a monohybrid cross. So yes, a recessive allele may not manifest its phenotype in one generation, but it can in subsequent generations. For example, cystic fibrosis. You could be a carrier, and then in the next generation, your children could have cystic fibrosis, depending, again, on independent assortment, random segregation. So, can someone tell me what is big R and what is little r on the DNA level? What is the difference between a big R and a little r at the genomic level? You learned dominant and recessive. Yes? Different protein coding genes. Okay, I think it's the same protein coding gene, the differences in them, very good. It would be the difference in sequence. The sequence is different. It could be a point difference. I don't want to say mutation, because another important point, recessive genes, okay, if I say this is a recessive gene, it does not work. Would that be a correct thing to say, that this recessive gene? Absolutely not. Recessive genes are not genes that are non-functional. They're always dead. They can't work. The recessive can mean two different things. Eye color, you can't say someone's eye color is not working, right? You just say this is a recessive allele. For a disease, you can say, yes, this is recessive because this gene is not producing a functional protein, for example, cystic fibrosis. The CFTR receptor is not working, which is not pumping chlorine properly, which is giving you a dry mucus. And so recessiveness does not always mean that the gene is not functioning. Just always remember that. So right, you're right. It's a difference in sequence. It can be a point difference. It can be a deletion. It can be an inversion. It can be an insertion. You learned all those mutations. All of those are differences in alleles. So this is dominant and recessive. What about codominant? See this flower here with the red and the white? That's codominance. Codominance means that both the alleles are going to manifest their full phenotype, both. It's not a competition. Who wins? They both win. So these cells here that are producing the red pigment are saying, we are going to produce red. And these cells here are saying, we're not going to produce red. So no one wins. So the white is not producing a white pigment. The white over there, the cells in the white petal are not producing red pigment. This is codominant. An example of codominance in our cells, in our body, is blood type. A, B, and O. So these are cell surface antigens on our red blood cells. And so if you have an A, then you only have A antigens. If you're type B, you only have B type antigens. But if you're AB, there's no competition. No one's winning. A and B are both going to be expressed on the cell surface of your red blood cells, so therefore you have both antigens. O has no antigens at all. Here, you see this pink flower. This is an example of intermediate expression. Intermediate expression. This means that neither allele will fully express its phenotype. Neither win. It's a compromise. You could see it that way. The cells that want to produce red will produce less red, and the ones that want to produce white will produce a little bit of red. So they're compromising. So now it's pink. There's no pink pigment. It's less of the red. So Mendel employed the scientific method through careful experimentation with controlled crosses and quantitative analysis. Controlled crosses. He kept one thing variable and the rest constant. Then changed the next thing and made that the variable and everything else constant. If you keep changing everything, then you have no idea what you're studying. Why is it that you're seeing the effect that you are? So by doing this, he said, you know what? There are factors that are leading to this phenotype. And those factors were, or was, DNA. He discovered, essentially, these factors are DNA. You know, imagine, guys, just think about it, right? I mean, he's able to deduce all this just by looking at peas. That's pretty impressive. I mean, at least I think it is. So what did he conclude? He concluded that variation in traits is due to different alleles. There's a difference in alleles, which gives you the different trait, i.e. big R, big Y, little y. Alleles segregate randomly into gametes. So here you have random segregation. This little r could have easily gone into this cell, and this big R could have easily gone into this cell. It's not predetermined which chromosome is going to go into which cell. Organisms inherit 2 alleles for each trait. This is a Mendelian form of inheritance, 2 alleles for 1 trait. Appearance of heterozygotes is determined by dominant alleles. Most of our traits is polygenic. We don't have a lot of Mendelian inheritance. A lot of our traits are not Mendelian. In other words, just 2 alleles are involved and boom, you have this trait. If that was the case, do you think we would have found the cure to every disease possible? We sure would have. 2 alleles, fix one of them. One of them is mutated, one of them is not. No, it's polygenic. Your skin color involves 150 genes, 150. They thought it was like 350, then some studies say 165, some say 135. The bottom line is average, let's say 150. 150 different genes come together to make your skin color. So most of our traits and our diseases are polygenic, which is the reason why we really have a hard time with finding cures for a lot of these diseases, because it's multifactorial. You have the genetic, plus you have the environment, all coming together. It's a hot mess. So here you have this diagram from your textbook. You can see this is meiosis in the male or female diploid parent, the diploid nucleus. Then you get DNA synthesis, you get homologous pairs of chromosomes. They separate, as you know, and then the sister chromatids separate, and they make the gametes. So just follow this through and understand how independent assortment and random segregation happens here. Okay, so if I were to ask you on the exam, this is going back to me caring about you guys again. This is what's happening. If I were to take this cell and tell you and ask you, what is the N value in that cell? What is the N value? 2. Not the coefficient of N, I'm asking you for the N value. Sometimes people go, oh, I thought you meant coefficient of N. No, don't think what I meant, know what I meant. I said N value, I didn't say coefficient of N. Okay, so what's the ploidy? What's the coefficient of N? 2, the ploidy is 2. And it has 2 copies of the genome, it's 2C. What is one set? This means that you have 2 sets of unique chromosomes. That's what the definition is. Is this one set here? No. One set is a big one and a small one. Don't worry about these 2 or these colors. First of all, the exam is black and white, so don't worry about colors. Take a big one and a small one. Big one and a small, that's one set. That's another set. All right, what if I said in this cell, if I take these 4 chromosomes, these are 4 chromosomes, not 8 chromosomes. If I take these 4 chromosomes and I weigh them, and they turn out to be 40 picograms in weight, what is the C value? What is the C value? Can someone tell me? What's the C value? Those are the options. 10? Anyone else saying something different? So we have a 10. Yes? 40? Guys, I don't want you to guess. You're not in a position right now to guess. You can do this. I have faith in you. I have faith in you. Yes, all 4 chromosomes, every DNA you see in the cell, every piece of DNA you see in the cell weighs 40 picograms. Yes. 20? What did you say? Yes. 4. Guys, I'm so glad I'm doing this. I love you. I'm so glad I'm doing this. Guys, what do you have to determine to know what the C value is? Remember, I said the secret sauce is the coefficient of C. What is the coefficient of C in this cell? 4. It's 4C equals 40, therefore C equals 10. The answer is 10. Next question. Circle one genome, one copy of the genome in the cell. Circle. So I won't ask you to circle some multiple choice. I'll give you 4 cells, and I'll have them all circled. I'll say, pick the cell that has the genome correctly circled. What would you see circled for the correct genome? Yes? Excellent. Good. Okay. Would it be this? No. Would it be this? No. Good. Would it be this? Excellent. We're getting there. Would it be this? No. That's where people go wrong. They're like, yep, that's one it. One big one, one short one. We're good. No, 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 no. One sister chromatid of the long, one sister chromatid of the short. That is one copy of the genome. 1, 2, 3, 4. This was a good exercise. Take this example as like a foundation and base, and then just work your way. This is the kind of exam question. Is this a good exam question? To understand if you know what the differences are between these things? Good. We did it. We did it. Okay. You studied for it. See, I studied with you. All right, monohybrid crosses. For this cross, you just have to go back to grade 1. Go back to grade 1 and look at the genotype, write them out, separate them, put one genotype in the horizontal bar, the other one in the vertical, make the square, and literally copy these numbers at grade 1. Big P, big P, big P, little p, big P, little p, little p, little p, done. Now switch back from grade 1 to university. Now, what is the genotypic ratio? Genotypic ratio is 1 to 2 to 1. 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. This is the genotypic ratio. What is the phenotypic ratio? 3 to 1, excellent. 3 purple and 1 white. I will ask you to calculate this stuff on the exam. And you guys know that one of the multiple choice questions is going to have both the genotypic and the phenotypic ratio in it, right? So understand what the difference is. The phenotype is like, what color do you see? What size do you see? What shape do you see? That's phenotypic. Genotypic is the actual genotypes. In other words, is it a dominant, recessive allele? Which alleles are those? So you go from monohybrid, now we have a dihybrid cross. So a dihybrid cross is explained beautifully in the textbook. Go and read it and understand it. A dihybrid cross gives you a 9 to 3 to 3 to 1 ratio genotypically. The phenotypic ratio will be different. It could be, but it will give you a 9 to 3 to 3 to 1. That's the genotypic ratio. The way you have to do this is to make the gametes and then line them up. And then you can write these all out, but there's also the shortcut version, which is right here. I have a video on Brightspace showing you exactly how to do a question like this. So you have to make the gametes first. Big R, pair it with another Y. Then big R with the next Y. Little r, big Y, little r, little y. Do not put two r's in one and two y's in one. That makes no sense for a gamete. That's aneuploidy. So you want to make sure that normal gametes will have different alleles. How do you calculate how many unique gametes are made when you're given a specific genotype? In this genotype, you have 4 unique gametes, right? The way to calculate is 2 to the power n, where n equals number of heterozygous pairs, not homozygous, heterozygous pairs. So how many heterozygous pairs do you see here? 1, 2. So this is why this is 2 to the power 2, which is 4, and you have 4 unique gametes. If I put capital M, what would be the answer? How many unique gametes am I going to have? You put M, capital M in all of them. That's still 4 unique gametes because you still only have 2 heterozygous pairs. This is homozygous. Don't add homozygous in the formula, only heterozygous. Okay, now what if this was the genotype? Then what? How many unique gametes would this individual make? 8, because 1, 2, 3 heterozygous pairs, 2 to the power 3 equals 8. All right, beautiful. Just remember that formula and you'll be fine. Polygenic trait, it shows a continuous variation. As we talked about, there are 150 genes that make your skin color. I bet you if we all lined up and compared our skin color, I bet you none of us, no 2 of us would have the same skin hue. Would you agree with that? I don't think we would. With 150 different combinations and how it could happen? Nah. The last topic to talk about is epistasis. Epistasis is a very interesting topic. You will encounter this in upper years. So let's go over the basics of epistasis. What is epistasis? Epistasis is a form of non-Mendelian genetics, just non-Mendelian. It's when one gene interferes with the expression of another gene. I can't tell you how much I learned epistasis over the years, and no one explains it to me properly. It's like, why is epistasis so complicated? It's not. It's honestly about how you explain things and how you learn things. Like, nothing is complicated. If you want to make it complicated, it becomes complicated. Epistasis is when one gene is interfering with the expression of another gene. That's epistasis, simple as that. Epistasis is a form of non-Mendelian inheritance in which one gene is capable of interfering with the expression of another. Here's an example. Gene A is involved in pigment production. It makes the pigment, so it can be hair color, eye color, it can be skin color, it can be the coat color, right, of an animal, color, pigment. Gene B has to deposit the pigment. It's not enough just to make the pigment. The pigment has to be deposited in the correct cells in order for you to see the color. So they both have to work hand in hand. So gene A here, big A, big B, you get pigment because both are happening. Pigment is being produced and it is being deposited. Big A being dominant and big B being dominant, you can see that even in this heterozygous form, they're going to produce pigment. Because again, pigment is being produced and it is being deposited. However, pigment here is being produced, but it is not being deposited because of the little b, so you get no pigment. And in this case, gene B is interfering with the expression of A. Here, you don't see any pigment production. Well, it doesn't matter that it's ready to deposit if there's no pigment to deposit. So if you don't even make pigment, there's nothing to deposit, so you get no pigment. A great example is the coat color of Labrador retrievers. Here you have 2 genes that affect the same trait. Big B makes black pigment, little b makes brown pigment. Big E deposits the pigment, little e does not deposit the pigment. If I were just to give you this information on the exam, would you be able to tell me what the coat colors would be based on genotype? You should be able to, if I just give you this top part. That's a hint. So let's look at this. You have a black Labrador here. Big B dash simply means that this can be a big B or a little b, or a big E or a little e, the outcome will still be the same. The phenotype will still be the same because B and E are dominant. This here gives you a chocolate Labrador because little b produces brown and big E is there to deposit the pigment. So brown pigment is deposited. In this case, you get a yellow lab because even though the pigment is being produced, both black and brown, you cannot get deposition of the pigment. So there's no yellow pigment in the yellow lab. It's just no pigment. If you take a black Labrador with this genotype and a yellow Labrador with this genotype and cross them, all the pups are black. So you must be like, oh, so this is like a dominant recessive thing, right? So what would your prediction be if you made it two black labs? If a black and yellow give you all black, then a black and black prediction-wise, you would say probably all black, right? But that's far from the truth. You get this phenotypic distribution. You get 9 black, 3 chocolate, and 4 yellow. This tells you that there's an epistatic event taking place because this is non-Mendelian. It's not 9 to 3 to 3 to 1. It's 9 to 3 to 4. Let's go back one more thing. 9 to 3 to 3 to 1. Write this out on the exam. And then you can just write these out and see what proportion have this, what proportion have that, and you can determine the phenotypic ratio. So this line simply means, guys, that this could be a big R or a little r, big Y or a little y, it will still mean the same thing, and vice versa for all of them. So this could be big R, big Y, little y. The phenotype will still be the same. So understand, this is epistatic event because of pigment production and pigment deposition. So on that note, I'll see you guys next class.
Dr. Sharon,